Welcome back. Today we are expanding our view to look at multi-step tumorigenesis and deconstructing the true complexity of cancer. This framework is heavily rooted in the seminal papers by Hanahan and Weinberg from 2000, 2011, and 2022, which have accumulated over 20,000 annual downloads. As illustrated in our progression model, the journey begins with a normal cell that undergoes an initial mutation, turning it into an initiated cell. Through ongoing cycles of accumulating mutations and passing through the central hallmarks wheel, a pre-cancerous lesion eventually develops into a fully malignant tumor. The first major hallmark is sustaining proliferative signaling, which acts like a gas pedal stuck firmly to the floor. Cancer cells achieve this through autocrine signaling by producing their own growth factors and via receptor overexpression that makes them hyper-responsive to external cues. Furthermore, intracellular circuit mutations lock these signaling pathways into the on position. For instance, RAS oncogene mutations actively drive this process and are found in 25% to 30% and up to 35% of human cancers. Next, cancer cells must evade growth suppressors by effectively cutting the cellular brakes. Normal cells rely on tumor suppressor genes as brakes to halt division during diverse stress, using DNA damage checkpoints to block the cell cycle. In cancer cells, however, these brakes are disabled, causing the cell cycle to run completely out of control and ignore normal tissue boundaries. Key highlights include TP53, known as the guardian of the genome, which is disabled in roughly 50% of cancers, as well as the RB protein, acting as a master gatekeeper. Ultimately, cancer cells mutate, delete, or silence these protective checkpoints to sustain their expansion. To survive genomic chaos and aggressive treatments like chemotherapy, cancer cells must resist cell death by impairing their internal self-destruct protocol. The cellular balance shifts decisively towards cell survival as anti-apoptotic shields and physical shields outweigh pro-apoptotic triggers. A prime example of this is the overexpression of BCL2, particularly seen in lymphomas. Fortunately, modern science has developed targeted therapies, such as the drug venetoclax, to combat this specific evasion tactic. Moving forward, cancer cells must enable replicative immortality by acquiring endless cellular fuel. Normally, as telomeres shorten with each division, cells eventually hit the Hayflick limit and stop dividing. However, the telomerase enzyme is reactivated in 85% to 90% of all human tumors. By looping and rebuilding these protective chromosome caps, telomerase allows cancer cells to achieve immortality and bypass the Hayflick limit entirely. In summary, looking at the broader picture, the framework has evolved from 6 original hallmarks in the year 2000 to a comprehensive map of 14 capabilities and enablers by 2022. Genomic instability acts as a core driver linking all of these interconnected cellular capabilities. While modern precision medicine targets specific individual hallmarks, such as using EGFR inhibitors, tumor plasticity, and adaptation mechanisms, mean that resistance often develops. Because of this dynamic resilience, tackling advanced malignancies increasingly necessitates multi-hallmark combination therapies rather than single-target approaches.